Hello, good morning, students. Today we will study next point from this chapter, biomolecules, that is denaturation of protein. So what is meaning of denaturation of protein? So at high temperature, then in the presence of acid or in the presence of base or by agitation can disrupt the non-covalent interaction responsible for specific shape of protein. This specific shape of protein is due to non-covalent interactions. So this process is called as a denaturation of protein. Actually, the denaturation is a process by which the molecular shape of protein changes without breaking amide or peptide bonds. So these are the covalent bonds. So they do not break, they do not changes during this denaturation, and they form the primary structure. This amide bond or the peptide bond that forms the primary structure. The secondary structure, tertiary structure, and quaternary structure, these are formed due to non-covalent bonds, and this interruption of this bonds, that is a non-covalent bond that take place, and therefore molecular shape of protein changes by without breaking this amide linkage or the peptide linkage. This is called as a denaturation of protein. Very good example of denaturation of protein is given here in the box. So here the globular protein, they are typically folded, they are hydrophobic, means no interaction with water molecule for the side chains in the interior part, and the polar residue on the outside, and therefore they are water soluble. Due to this polar residue in the outside, this globular proteins are soluble in water. Now, due to denaturation, this exposes the hydrophobic region of globular protein, and therefore this globular protein becomes water insoluble. So how this change is observed, that is given in the shape, that is coiled globular protein. This is a coiled globular protein, and this coiling is observed due to this non-covalent bonds, that is the hydrogen bonds, van der Waals force. All these forces we had studied last time, which are responsible for secondary structure, tertiary structure, and quaternary structure. Due to this denaturation, these bonds breaks, and only this amide linkage or the peptide linkage is present. So this is a loose coils and the loops of the protein molecule that contains the amide linkage or peptide linkage. So here what happens after denaturation of protein, that is given here. So denaturation results in the disturbing the secondary structure, tertiary structure, and quaternary structure of protein, and this causes the change in the properties of protein, and biological activity is also lost. So that is the important effect observed after denaturation of protein, and that is about the denaturation of protein. Thank you.